Okay. So our next speaker is going to be Calvin Williams, who's in the MD PhD program at the University of Maryland in Baltimore, where he's in the microbiology and immunology department.
Good morning. The purpose of the talk, I have two primary purposes for this talk. One, I'm going to give you a brief overview of my thesis project and also show you the rationale behind my thesis project. Okay, hear me better now? So the title of my project is The Identification and Characterization of Plasmodium Genes Involved in a Protective Pre-Erythrocytic Immune Response. So basically what I study is a parasitic disease called malaria. And shown here on this map is the global distribution of malaria. It's primarily transmitted, since it's transmitted by mosquitoes, it's primarily in subtropical areas, and it accounts for 500 million acute cases a year and 1.5 to 2 million deaths a year, primarily in pregnant women and children under the age of 6. So malaria is caused by a parasite called Plasmodium, which is injected in the skin of humans or other mammals during the taking of a blood meal by a female mosquito. The parasite migrates rapidly from that site of infection to the liver, where it invades the hepatocytes, which are the primary cells in the liver. It undergoes some structural changes, and changes form from sporozoites to what are called merozoites, which are able to be released into the bloodstream where they can invade red blood cells. All the pathology associated with a malaria infection comes from the sequential infection and rupturing of large amounts of red blood cells. The only known vaccine to ever work in humans was to take infected mosquitoes and irradiate them and then allow those to feed on humans. Those parasites, called radiation attenuated sporozoites, are able to migrate to the liver, but they fail to develop inside the hepatocytes. They just remain persistently there, and they're associated with protective immune responses, which are primarily mediated by CD8 T cells. Unfortunately, this vaccine can't be used on a large scale because it would require billions of mosquitoes, and you have to be bitten by over 1,000 mosquitoes in order to have the vaccine work. So for obvious reasons, it can't be used. But it does provide a useful model, because if we can understand those antigens that are expressed by the irradiated parasite, we can make what are called subunit vaccines, which can be used in large numbers of people. So in order to understand, since I don't know what the antigens are, I needed to understand where the antigens, those protective antigens, would be expressed, so I don't know where to look during the life cycle, since it's a complicated life cycle. And in order to have an immune response, a protective immune response, you have to have activation of T and B cells, and then those T and B cells have to go and target something. So the activation appears to occur both after infection with wild-type sporozoites as well as the radiation attenuated sporozoites. So that leads me to know that the pre-erythrocytic stage antigens are expressed by those two types of sporozoites. Also, the target of the immune response is the early infected hepatocyte. So I know, again, another location where the parasite is expressing protective antigens. So that leads me to the hypothesis that the pre-erythrocytic stage antigens that are protective are expressed by wild-type sporozoites, radiation attenuated sporozoites, as well as early liver stage parasites. So the specific aim of my project is, one, to try to identify those targets by doing what's called a transcriptional analysis. What that means is I'll try to identify all or a majority of the genes that are expressed at each of the different stages of Plasmodium, and then try to characterize which of those differentially expressed genes have a role in a protective immune response. In order to do that, I'm going to use a model called Plasmodium uelii. It models Plasmodium falciparum, which causes the most mortality and morbidity in humans. And what I'm going to do is I'm going to take irradiated Plasmodium uelii sporozoites and extract the total RNA from those. That's all the RNA I can get from them, and do what is called a microarray analysis. And the microarray, what it does is it has each gene in the genome of Plasmodium spotted onto it, and then I'll hybridize the total RNA. So those genes that are expressed by the irradiated sporozoites will glow one particular color, and those that are not will not show up. So they'll let me look at all the genes at once instead of looking at them one at a time. I'll do the same thing for non-irradiated Plasmodium ULAI sporozoites. And then I'm going to take some of these non-irradiated sporozoites and infect biopsy mice. And those biopsy mice I'll take and harvest their livers and use a technique called laser capture microdissection, where I'm able to cut specifically the parasite out of the hepatocytes and extract RNA from those and also put those on the same array. So that'll give me information from each of the stages that I'm interested in. So what it'll do is it'll give me a zone here represented by the blue circle. I'll get all the genes that are expressed by radiation attenuated sporozoites. I'll get all those that are expressed by the wild type sporozoites. And right now I have a set of data from the genes that are expressed by the early 24-hour sporozoites, as well as those that are expressed by 48-hour sporozoites. And so far, these only show genes that are upregulated 2-fold or greater in each of the time points. So I have about 278 genes that appear to only be expressed by the 24-hour non-irradiated parasites. I'm on my way to actually do these. I do the arrays in Philadelphia, so I collect the RNA here, and I'll be doing the 2 arrays for the sporozoites in the next 2 months. So I'll have data for those 2. And hopefully what I'll be able to identify is a set of genes that are upregulated in the early liver stage by both radiation and non-irradiated sporozoites. And for those, I'll have targets that I could put into my DNA vaccine vector and use that to try to figure out which ones are actually immunogenic and which ones aren't. And that's basically what I just said. So I won't repeat that. So I thought I was going to talk slower, so that's all I put in my talk. So we have room for a lot of questions if you have them. That's it. Yeah.
I was just wondering why you picked the 24 and 48-hour time point. Is malaria somehow, does it leave the liver after 24 to 48 hours normally, or?
In mice, it stays in the liver for 72 hours. But the reason why I picked the 24 and 48, at 24 hours, it's still developing into what are called myrizoites. So it's kind of like what a caterpillar does when it goes into a cocoon. It goes in as a caterpillar, but it comes out as a butterfly. But while it's inside the cocoon, it's neither caterpillar or the butterfly. It's somewhere in between. So at 24 hours, the immune response, the T cells, they target those parasites that are within the parasites within 24 hours, but at a later time point, at the 48 hours, they are no longer targeted. So I know the protective antigens have to be expressed early, but not late.
You have a question over here? Okay. Sort of face this way.
Okay, so the vaccine that it takes 1,000,
Yeah.
how far out is the protective effect from that vaccine?
I think the longest, what they did was they did trials using people in the military, and they were able to re-challenge them up to 60 weeks after, and they were still protected. So that's roughly 6 months. And they did a few where they did it like a year or 2 later, but it wasn't statistically significant, a large number of people, to be able to say you can protect everybody for that long. But they've done it in a small number of people.
Another question? Somebody said something back. Stand up.
I think that's it.
Wasn't there another question?
All right.
So you said that the radiation attenuated sporozoites, those, for some reason, you can't use that because you need like so many.
You just needs too many, yeah, it's true.
I don't understand why. Would you be able to extract or figure out what it is that you need from them, and then maybe like you do in molecular biology, throw it up and then use it? Or?
That's what I'm trying to do by doing the RNA analysis. Okay, yeah. Sorry. Okay. That's it.
So is malaria, if I recall correctly, and I could be wrong about this, is one of these organisms that has multiple ways of changing its antigens?
Yes.
So do you have any, so the problem is that you make a vaccine against one antigen, and then the organism downregulates that antigen, and so you have great immunity, but it's not against your malaria agent anymore. So do you have any concept of how many genes you're going to have to express, or how many antigens you want in a big combination vaccine in order
Well,
to
the thing about usually for vaccines that have the problem you just mentioned is usually based on antibodies, because it's usually based on some protein that's expressed on the surface that's downregulated, and then something else takes its place. But the immunity for malaria is T cell mediated, and most of the genes that we hope are going to be used are going to be something that's conserved. That's what we're hoping. It's going to be something that's conserved, something that's not expressed on the surface, because it has to be degraded and expressed on the MHC. So it's not usually something on the surface. That's what we're hoping. But we also realize that since there's so many different stages in a parasite life cycle, you have to have genes from each of the different stages. Otherwise, even if we get 99% protection, preventing them from coming out the liver, if one gets out, then they'll just keep replicating in the blood stage, and we won't have any immunity to that. So we have to have something from the liver, something from the blood stage, and maybe even something from one of the mosquito stages also.